Okay, so let's go ahead and keep talking about the features of animals, what makes animals different than other groups we've discussed. Last lecture, we were discussing the developmental process. We left off with the developmental process. Starts with fertilization, egg and sperm combine. You now have a diploid organism or sporophyte, or we call it the zygote. It's all the same thing. Hox genes regulate the developmental process. You're going to divide, you're going to divide, you're going to divide. You keep dividing. 2 go to 4 to 8. We get the blastula forming. The blastula forms this ball, this hollow ball. The blastoceles on the interior of that. And as it keeps developing, the blastopore formation will determine or distinguish the difference between protostomes and deuterostomes. Now, protostome and deuterostome are not classification levels. They're not kingdoms, phylums, anything like that. They're simply developmental groups. So if you develop this way, you're a protostome. If you develop a different way, you're a deuterostome. So it's a general grouping of how we organize the animals. It's analogous to gymnosperm and angiosperm in the plant world. Those were not classification levels. Neither are these. It's just how we can kind of group the animals together. Will these become classification levels at some point? Who knows? That's a good question. That'd be a great question. It'd be a great project for you guys to think about and possibly explore. So, okay, so we got our blastopore forming, or blastula. We get the blastocele. Then we get into this stage known as gastrulation. Now, gastrulation is when the blastula, it starts to indent upward. It pushes inward, and it creates a hole. So the hole, this opening right here, is officially known as the blastopore. So think about pore. If you have a pore in your skin, that's a hole. That's an opening. So that opening starts to form. Now, depending upon which group you're in, protostome or deuterostome, you either stop right there, you got your pore, and then you have that kind of pouch or bag on the inside, right there, and you're done. Or it's possible that this opening continues to grow and develop and comes all the way out the other side. That's the difference between protostome and deuterostome. So if the blastopore becomes the mouth first, then you are considered a protostome. If the blastopore becomes the anus first, then you are considered a deuterostome. So what we'll see is protostomes, and this, a lot of animals are protostomes. They develop the mouth first. Deuterostomes develop their anus first. Echinoderms, chordates, all of your mammals, your reptiles, your birds, your fish, your etc. We're all deuterostomes. That means our anus develops first. Things like mollusks, worms, etc., their mouth develops first. So seems a little ironic, but that's just how development works, the developmental process within the animal world. Okay, so re-emphasizing. Notice the stress in the hint here. Protostome, mouth first. Deuterostome, anus first. Sometimes you don't even develop this complete digestive cavity or tract. It doesn't go from mouth all the way out through the anus. It stops, like what we saw up here, and just stops as that, excuse me, that type of pouch there. So we'll see some variation again as we go through the different animal groups. So that is one feature we're going to use to identify the difference between protostomes and deuterostomes. So here's a little hint, not a little one, it's a big one, should be a big hint for you guys. I want to try to be very, very fair and upfront. Okay, let me what I want you to start to do is create a list of features okay. So create a list of features get the hint that distinguish protostomes from deuterostomes. How do we know the difference? What are the key characteristics? So if you had to list out 3 or 4 features that said, ooh, this is how I know it's a protostome. This is how I know it's a deuterostome. Do it. Make sure you guys get this list down. So right now you have one feature. What develops first, your mouth or your anus? Second feature, as you're going through cleavage, let me get that out of there. How does your body plan form? So cleavage is the divisioning of the cells. 2 divide into 4, 4 into 8, 8 into 16, and so on. We'll see 2 different patterns of cleavage. Radial cleavage is found among the deuterostomes. Spiral cleavage is found among the protostomes. So there's your second trait or feature. So when we talk about radial cleavage, imagine having a line or a shaft or a rod or whatever straight up and down, and then as the cells are dividing, they're just stacking up one level on top of the next, on top of the next, around that central axis or shaft there. That is radial. Spiral cleavage, same process of cells dividing, but as they divide, they start kind of stacking up in a spiral pattern. This is not a really good way to draw it, but I apologize. I'm working with the tools I have here. The cells stack up and they kind of twist in a spiral pattern. Think about a spiral staircase or a corkscrew. That is spiral cleavage. So deuterostomes, radial cleavage, protostomes, spiral cleavage. Another feature that helps distinguish protostomes from deuterostomes. Okay, so we want to make sure we're comfortable with those differences. All right, we're almost at the end here, so hang on, just a couple more slides to go. So, okay, other traits of animals we want to look at. Segmentation of the body plan. So, is your body made up of different segments, different body parts, different pieces? That segmentation of the body plan is directed by master control genes called, do a drum roll, who knows this? Hox genes. Those famous Hox genes we talked about earlier, those are the ones that dictate and control segmentation of your body plan. So think about an earthworm, all those different little segments to that body plan. That is dictated by the Hox genes. Wasp, same thing, different segments, different parts of the body. You have a head, a thorax, an abdomen. All that is controlled by Hox genes. Now what's crazy and getting really cool to understand, the Hox genes will determine, okay, here's your thorax, chest region of this wasp. There's a gene that says, okay, let's produce a wing here. Up in your head region, let's produce an antenna. There are genes, the instructions that determine this. We can tweak these genes, and mutations do it unintentionally, but it happens. But we're learning how to tweak them where, I don't know why you would do it, but you can tweak the gene to produce a leg coming out of the individual's head. Now, definitely not a good feature for the organism. Do you tweak it to produce an extra set of wings off of the thorax? It's all being controlled and dictated by those Hox genes. So our knowledge is getting better. And for those of you excited about the molecular field, look into these genes. There's so much we don't know about the Hox genes and so much we still can learn Who knows what we'll do in the future with this kind of stuff. So, all right. So Hox genes going to play a role in determining all this information about the animals and other groups as well. So, okay. So in the big picture, we have this huge diversity of animals on this planet. Environmental influences, temperature, sunlight, climate, moisture levels, all those non-living factors, environmental factors, along with evolutionary processes, have helped produce this diversity of animals on Earth. So look at the spectrum of animal diversity. It's broad, it's vast. Well, animals have been evolving for hundreds of millions of years, and this is what has led to the diversity we have today. So the last thing I'm going to leave you with here. Oh, sorry about the spelling. Let me get that corrected. All right, so here's your $100 question. You get it right, somebody in the class will give you $100. Oh, wrong thing, sorry. What is the ancestral group that gave rise to the animals? Oh, too much. Gotta take the bold off, sorry. All right, what's the ancestral group? So, which group did we talk about in previous lectures that are the ancestors to the animal kingdom? I want you guys to see if you can figure that one out before you jump to the next lecture. So this is it for, excuse me, for our animal diversity lecture, the chapter on animal diversity. The next lecture is going to be a bunch of small lectures over the protostome diversity, and I'm doing it in small bites to make it easier to tackle. But this chapter, this animal diversity basic content, is laying the foundation for all the animals that we're going to discuss over the next several lectures. All right, so check it out. Fill in those blanks. Make sure you guys are taking notes. Don't just be a passive student sitting here and just listening. Take notes. Write them out. Answer some of these questions. It will make getting through this material a whole lot easier, and you're going to absorb it so much better if you're actually writing this stuff out instead of just watching the video and listening to the video. Okay, so next lecture, we'll get into protostomes and all of the different groups that are organized under the protostome category. Remember, protostome, I want you to know the features of protostomes, the cleavage pattern, the developmental pattern, etc., how we get to a protostome, and protostomes are not a classification level, it's just an organizational level. So we'll start up with those guys in the next lecture.